Hi, everyone. Welcome to today's webinar. My name is Christy. I'll be moderating today's webinar titled Confidently Validate Cancer Models for Precision Medicine. We have our esteemed speaker, Dr. Robert Bowman, Assistant Professor at the University of Pennsylvania, presenting along with our product manager, Divya Rao. Dr. Robert Bowman will be presenting on the modeling clonal evolution in myeloid malignancy, and Divya will be giving an overview on the Tapestry single-cell multiomics technology. If you have questions, please submit it into the Q&A box. To find that, it's at the bottom of the window. Click on it and submit your questions. We'll address those at the end of the webinar. If there are any technical difficulties, please email into info@missionbio.com. And without further ado, I pass it over to Dr. Robert Bowman.
Okay, thank you so much, Divya, for the introduction. Today I'm going to share with you all some of the genetic and genomic approaches we've used for modeling clonal evolution in myeloid malignancy. So to kind of kick us off, let's discuss, first off, how we think about patterns of somatic evolution in cancer. Generally, we think about these as ways in which clones of cells grow out over time. So we can think of this as kind of a Darwinian evolution process, right? Where a cell is, stem cells sitting there, picks up a mutation, and it divides. And one of those two daughter cells may have a fitness advantage, and that leads to it dividing either a little faster or existing through stress a little better than the other cell, right? And this can happen neutrally over time, where maybe there is no fitness advantage. You just have mutations kind of percolating through a system, or cells can be selected for, and this leads to clonal expansion. It can happen in a very linear pattern where cells pick up mutations slowly over time, one step at a time, and they get bigger and more fit. Or it can happen in a very branched manner, right, where cells, certain subclones are picking up different mutations and leading to different fitness evolutions through time. And this can be a gradual process. Think about mutation patterns that happen with aging or constant carcinogen exposure, and slow acquisition of new mutations over time. It can be a heavily punctuated process, perhaps large exposure to a huge amount of carcinogen or a major stress event that leads to the change in evolutionary landscape. So we want to understand these processes, how they relate to cancer development, and how that kind of leads to progressive cancer development. So I've talked about mutations. There's a lot of different types of mutations you could consider. We can think of numerical chromosomal abnormalities, maybe a trisomy or a loss of a heterozygosity of allele. We can think about chromosomal instability, where perhaps you have translocations. The majority of what we're going to think about today actually lies in the somatic mutagenesis, where we have single nucleotide variations. And we're going to focus on those a lot today. And that's not to say that there's not a lot of epigenetic heterogeneity, right? Where a cell can be locked into a specific state, and that might be affecting differentiation. That's something we're totally interested in, but today we're going to focus really on just somatic mutagenesis. Now, one of the earliest forms of discovered somatic mosaicism was in the peripheral blood, the process known as clonal hematopoiesis. And a lot of this has to do with the idea that sampling peripheral blood is pretty straightforward. You can get a lot of different samples from a patient. You can track them over time, and they're able to be sequenced over time. However, as more and more tissues have been studied, somatic mosaicism has been really found to be an ever-present event. That's true in many different organs, true in the esophagus, the liver, the blood. We think this underlies a lot of clonal evolution in many different cancer types. Our focus on hematopoiesis really has to do for the ease of sample access, right? We're able to study this over time in patients, but this is a very experimentally tractable system where with decades of research, we understand the immunophenotype that's centric to different cells in the hematopoietic hierarchy. So we can go in and flow sort out or identify with proteins what a hematopoietic stem cell is, how it differs from a whole class of progenitor cells, and how those cells differ from terminally differentiated executor cells with distinct functions. So just the wealth of knowledge about this hierarchy lets us understand which cells are expanding during disease, and lets us build models where we can actually turn on mutations in these different cell types. So if somatic mosaicism is everywhere, clonal hematopoiesis is a really tractable system to let us study it. And one of the key pieces that's really helpful is being able to transplant these cells. So we can make dense genetic mouse models with many different alterations, take one mouse with that allele space and expand it massively into 50 or 60 different mice. And this lets us have the sample size needed to do extensive preclinical testing, or be able to do complex genetic processes that really can't be done otherwise. So here we're going to refer to, right, somatic mosaicism as clonal hematopoiesis, and we're going to be talking about somatic variants in that setting. So what are those somatic variants? The two most common mutations in clonal hematopoiesis are in the DNA methyltransferase DNMT3A. And when you DNMT3A, this largely gives a hypomethylation genotype. The second most common mutation is in TET2, which when mutated often gives a hypermethylation phenotype. So obviously in many forms of clonal hematopoiesis, DNA methylation is very centric to this process. So not only are these the most common mutations in clonal hematopoiesis, they're also some of the most common mutations in acute myeloid leukemia, one of the lethal consequences of clonal hematopoiesis. Now, clonal hematopoiesis itself is pretty common. Over the age of 60, most people, if you sequence deep enough, will have evidence of clonal hematopoiesis. That is, 30% of the population may have variants in their blood over 2% of variant allele frequencies. This is a lot of people. Yet acute myeloid leukemia is extremely rare, right? Not many people end up having acute myeloid leukemia. So, the reason I have my lab and what we want to understand is why is this process so common in clonal hematopoiesis and why is this process so rare? And what are the pressures that are driving this clonal evolution over time, right? So we have a good idea of which mutations occur in AML and acute myeloid leukemia, and we have an idea that the combinations really matter, right? So if you have a NPM1 mutation, a DNMT3A mutation, and a FLT3 mutation, this leads to really poor prognosis for patients. And if you don't have that FLT3 mutation, you have a better prognosis. So we know that combinations of mutations can inform prognosis. And bulk sequencing studies have given us an idea of how many different clones are in a patient, right? If we have, 2 to 3 different mutations or 4 or 5 mutations, how many distinct, clones do we have? And the answer seems to be maybe 2 to 3 big clones and maybe a lot of smaller ones. So with these ideas in mind, the third thing that informs our understanding of leukemia genomics is the order in which these mutations occur. And we can infer that through variant allele frequencies or how much of a sample is actually mutated. So DNMT3A, for instance, is often found with a variant allele frequency of 0.5, suggesting it's potentially heterozygous in every single cell of a leukemia if it's mutated, or potentially there's a large number of or homozygous cells in that sample. Meanwhile, if you have mutations in signaling genes like KRAS or FLT3 or NPM1, for instance, is another class of genes, these tend to have lower variant allele frequencies, suggesting they're later hits in leukemia and often present in smaller subclones. So, with this information, we start to get an idea of which genes are mutated, perhaps in what order as well. And this is kind of a global look at it. If we think that clonal hematopoiesis is premalignancy, what we really want to model and understand is the process by which we have this somatic mosaicism that is happening to lots of people, right? How does it become a lethal disease and how does it change over time? And when we apply different treatments, how is that disease going to further respond? So one of the tasks of my lab is to understand this process through the use of a couple different layers of experimental systems. One, we strive to enumerate clonal architecture with single-cell DNA and multi-omic sequencing, trying to understand which cells have which mutations. We've constructed a library of inducible leukemic alleles that are controlled by different DNA recombinases. I'm going to run you through some of those models today. Some of these models let us look for oncogene dependency, in particular FLT3 is the main gene we've looked at there. And then lastly, we have some nice ex vivo systems that we use to treat these leukemic cells with different small molecules or CRISPR-Cas9 editing to understand how these leukemic clones respond to different genetic or chemical perturbations, and then we're able to put them back into the mouse to kind of understand exactly their stemness. So the main engine of genetic manipulation in our lab are these DNA recombinases, largely based off CRE. These can lead to DNA inversions or deletions, depending upon the orientation of these LOXP sites. So if you have 2 LOXP sites pointed at each other, this leads to an inversion. If they're pointing in the same direction, it leads to a deletion. And this is a pretty well-studied recombinase. They'll be familiar to many of the mouse modeling field. The 2 other recombinases we use are FLIP recombinase and DRE recombinase. And the great thing about these 3 recombinases is they're all completely orthogonal to one another, and there's no cross-reactivity between them. So here's a nice little schematic that if we have a CRI reporter and we give CRI, that we only get recombination with CRI, not DRI or flip. And likewise, if we have a DRI reporter, we only give recombination. We only get recombination of that reporter when we give DRI and say with flip. So we're able to integrate these tools together now to study clonal evolution. So I want to show you how we've done some of that. The test case we're going to focus on is the receptor tyrosine kinase FLT3, and it has a lot to do with this effect on prognosis, right? It's the most commonly mutated gene in AML, frequently subclonal, like I mentioned, and it's never found in clonal hematopoiesis. This is a mutation that needs to be modeled late in disease development to reflect the human biology, and that's what we want to do. So there are existing mouse models of FLT3 mutations. However, they were constitutive models that were on since birth of the mouse. They do get, kind of a myeloproliferative phenotype. We wanted to engineer an inducible version of this model. So we took this well-established FLT3 mutant model, and we have now engineered that mutation in an inducible manner here. So what we did was we flanked it by FLP binding sites that led to, when FLP was added, this led to an inversion, deletion of the wild type exons, and expression of the mutant exons. And you can see a nice PCR here that when we give FLP, that leads to an excision of the event. We have an increase in this band size that we expected, and we're able to recombine this in about 50% of cells when we give tamoxifen to a FLIPO-inducible or tamoxifen-inducible FLIPO-ER22 allele. And this is nice. We can certainly titrate this tamoxifen down to give a more subclonal or a smaller mutant burden into the mice. And I'll kind of just move over this quickly now, but we've also engineered a DRI-inducible version of this allele, and that'll come up in a minute as you see how we can combine these different alleles together to model clonal evolution. So what happens when we take this allele and actually activate it? What we can do in the transplant system for hematopoiesis is we can compete it against 45.1 wild type cells. So we'll have our C45.2 FLT3 mutant cells or C45.1 wild type cells. We'll mix them together. We'll let these engraft into irradiated mice, and then we can give tamoxifen to activate the FLP-inducible FLT3 allele and track the ratio between mutant and wild type cells over time. And what happens when we do that? When we give tamoxifen, this leads to a large outgrowth of C45.2 positive cells, showing that FLT3 can lead to an increase in fitness of a clone, and it can be stable and really take over against wild type cells over time. And this is what happens in this primary transplant setting. Now, in the hematopoietic system, it's very common to be able to get outgrowth, but knowing if you really enabled a stem cell to kind of form leukemia and get a really disease on the mouse, the key trait is whether those cells can then continue to transplant to secondary recipients. So while we knew FLT3 could outcompete here, we needed to do a secondary transplant to understand if we've really elicited leukemia. So we took these cells, transplanted them again. In this setting, you could see that those cells were insufficient to reestablish disease and really were able to elicit proliferation, but not long-term self-renewal. And our thought was, well, maybe that's because these mutant cells need a clonal hematopoiesis allele or some mutation beforehand to really model full-blown disease development. So what we did was we crossed this mouse. We crossed this mouse with other alleles, and along the way we started to understand, why is this FLT3 mouse failing? Why is it not really giving rise to disease? We were able to pin this down to loss of specific transcription factors involved in hematopoietic regulation. So when you activate FLT3, you lose the expression of critical factors such as MECOM or EBI1, HOXB5, MEIS1, TAL1. All of these transcription factors seem to disappear. So how are you ever going to turn this thing into a leukemia? So we worked to combine it with two of the most commonly mutated genes in AML. One, we combined it with a FLP-inducible NPM1 model that was developed by Jen Trowbridge. So here we can cross these mice together, transplant them into recipients, treat the mice with tamoxifen, and that will lead to the excision of both alleles, turning on both NPM1 and FLT3. We can also do this with a layered model with a CRI-inducible DNMT3A. And in this setting, we can isolate hematopoietic stem progenitor cells from a double mutant mouse, electroporate CRI mRNA to turn on the DNMT3A allele, and then hit those mice with tamoxifen to turn on the FLT3 allele. And this is what we saw. The FLT3 NPM1 combination gave rise to disease within 5 weeks. And these mice were uniformly lethal, and the disease was 100% penetrant. The DNMT3A FLT3, this really didn't have much of a change in disease penetrance compared to FLT3 alone. In fact, these mice, they looked a lot like the FLT3 alone mice. But critically, when we went to transplant these FLT3 DNMT3 alleles, now these cells could actually engraft into secondary recipients and give rise to disease. And this is the idea here, is that DNMT3A seems to be licensing FLT3 for malignant transformation. And the latency of it is quite different than FLT3 and PM1. So this gives us an idea of how these alleles combine together to give rise to disease, and the latencies that change when different alleles combine together. So I've mentioned, that's one way that we've gone about modeling AML. We would also like to have ways where we can separate out NPM1 and FLT3 and do those sequentially as well. So I'll use this as a chance to highlight our DRE-inducible FLT3 allele. Here we crossed our DRE-inducible FLT3 allele with a CRI-inducible NPM1, so a different NPM1 allele. We can use a tamoxifen in this setting to activate the NPM1 allele, and that will turn on NPM1 here in all cells in the mouse. We can isolate the lineage negative cells, or the hematopoietic stem progenitor cells. Here, electroporate with DRE mRNA and then transplant. And you can see that this gives rise to disease again at that same 5-week frequency. So just kind of another way that you're able to induce this model. And this gives us a lot of exciting opportunities for studying things like cell of origin, where we can start to put this DRE mRNA into different cell types and understand which cell types are competent to transform when we activate FLT3. And this just gives you a flavor for how we do that model. So, we need to be able to move beyond some of these different transplantation models. We need other ways of turning on recombinases beyond just the tamoxifen-inducible FLT3. So one way we've gone about doing that is making inducible DRE alleles, right? So this DRE allele that we developed was inspired by other approaches that have been used in Cas9, where we took an NS3 protease and put it right in the middle of DRE. And here, this is autocatalytics. This will lead to the separation of these two halves of DRE. And right next to the cut site, we put a degron, so that as this is translated, the NS3 protease cuts and the N-terminus of DRE is degraded. If you use this protease inhibitor grazoprevir, you can prevent that proteasome or this proteolytic cleavage and keep an entire complex together. And this works quite well in comparison to CRI-ER, for instance, and ex vivo recombination. We're able to get highly specific recombination of a fluorescent reporter and activation of DRE stable. And this did not cross-react with the tamoxifen-inducible CRI-ER. We went ahead and we made 2 knock-in mice for this allele. So we knocked in this inducible DRE either to the VAV1 locus or off a strong synthetic promoter at the H11 locus. And if we dose these mice in vivo with the NS3 inhibitor, you can see that we get a high amount of recombination with a strong synthetic promoter and kind of a low level of recombination with the VAV1 locus. So we have, the drugs are bioavailable, we're able to get them into mice, but depending on where you put them, you get different levels of recombination. So in many ways, this VAV1 knock-in, it's not very inspiring, right? 0.1% of cells. But if you think about how much allele burden is necessary to give rise to an AML, this becomes an exciting opportunity to actually try and dial this down and see how subclonal can we really start this disease. So that's what we did, is we took this VAV1 DRE and we crossed it in again. So this is a little more of a complex model, but here we are. We have our CRI-inducible NPM1 or IDH2. We have a DRE-inducible FLT3. And now, in the absence of transplant, we can use tamoxifen to activate NPM1 or IDH2. Let this sit for a little bit. We can certainly vary this amount of time to ask questions about how much time between mutations is necessary to give rise to disease, but that's kind of the power of having these on separate recombinases. And in this setting, now we can give the protease inhibitor GZB to activate FLT3 mutations, and then we can see if this kind of recapitulates a lot of our transplant phenotypes. So when we do that, looking at kind of monocyte counts in the peripheral blood, we'll see that FLT3 alone doesn't really change these monocyte counts. FLT3 IDH2 leads to an increase in monocyte counts that kind of stabilize and smolder over time. And here, once again, our FLT3 NPM1 model leads to 100% lethality within 6 weeks. And the difference here compared to other models is we're doing this in a transplant-free setting. And we're initiating FLT3 mutation burden at 0.1%. It's a very, very small percentage of cells are actually mutated, but they're able to grow out and give rise to disease. Right? As we build this kind of catalog of different inducible alleles, I just wanted to highlight different ways we can use them to look at clonal evolution in mice. So here's a kind of one last experiment I'll show where we looked at wanting to model the sequential acquisition of DNMT3A, then NPM1, then FLT3. And through the power of multiple rounds of transplant and hematopoiesis, we're able to model this triple mutation and every other combination we want from a single donor, right? We can take cells and split them into whether they receive CRISPR-mRNA to activate DNMT3A or not. And then we can split those mice and give them tamoxifen to activate NPM1 or not. So we go from one donor to 4 different experimental groups in one transplant. We can then do a secondary transplant and give these mice DRI mRNA and activate FLT3 or not. And in the end, we end up with another set of 4 allele groups. So for one donor, we're able to look at now experimentally wild type cells, DNMT3A mutant cells, NPM1 mutant cells, NPM1 FLT3 double mutant, the DNMT3 NPM1 double mutant, and the triple mutant. And this really highlights the power of doing synthetic biology approaches in a model system that can be transplanted. So does it matter? Do we see differences? We certainly do. NPM1 FLT3 is the fastest to give rise to disease. And actually the addition of a DNMT3 allele seems to slow down that disease a little bit. That's true if you have FLT3 or not. And immunophenotypically, we see that the addition of FLT3 leads to an increase in our stem cells, this lineage negative, SCA1 positive, KIT positive stem cell, LSK population in mice. And this is especially true if you have that early DNMT3 allele, this kind of branch here. This leads to the largest increase in stem cells. So you don't necessarily need a clonal hematopoiesis mutation to give rise to disease, but it does change the phenotype downstream. And this highlights some of the questions we're trying to understand now. How does clonal hematopoiesis set the stage for AML? So we are very happy to get the result and see that this kind of clonal evolution model gives rise to differences in cell types. We needed a way to be able to know if this was relevant in patient samples and in human biology. So we turned to using single cell DNA sequencing via a tapestry platform. And this is something we've had experience with previously during my postdoc in the Levine lab. I should say much of this work was done during my postdoc in Ross Levine's lab. This is a model that we're quite familiar with. It's a single cell encapsulation model, and the custom platform or the custom panel we used here is the clonal evolution panel done with Lindy Miles as well. So we looked at 4 different patient samples, and this is kind of a common way that we look at the data called a clonograph, where the different color of the grid here indicates the mutation type. So heterozygous mutations here in NPM1, FLT3, and DNMT3A are the main clone in this sample. And there's a subset of cells that might lack that DNMT3A or might lack an NPM1. This sample here is MSK149. You can see the major clone here is actually DNMT3A TET2 mutant co-mutation. And there's a couple different FLT3 mutations that fall along down the line. We have a patient sample that's largely NPM1 mutant, where the NPM1 FLT3 clone is smaller. And then finally, we have that triple mutant we were looking at as well, another version of it where it's DNMT3 NPM1 and a bunch of different FLT3 mutant cells. So we had these, the next thing we needed was protein data. So with the DNA plus protein approach, we could take a look at a series of different protein markers. Here, this is a 45-plex assay, the total seq-D from BioLegend. And we were able to aggregate this data together across patients and take a look at what the immunophenotype of these different clones were. So one of our sanity checks in the data was to see if NPM1 mutant clones express CD34 or not. Most cases of AML with NPM1 mutation tends to be CD34 low, and that bared out in our data where the NPM1 mutant clones had less CD34 than the clones that were NPM1 intact. We're able to aggregate this data together and call different clusters of cells, and then look at the immunophenotype of those different clusters. So you can see our cluster 1, for instance, is high for FLT3, high for CD34, high for CD117 or KIT. Meanwhile, our cluster 2 was higher for differentiation marks like CD14, CD11B, and CD64. And then finally, our cluster 3 had more CD45 RA and CD38 expression, suggesting it might be more of a progenitor-like cell. So with that information in mind, we're able to kind of project these marks onto this UMAP and get different regions of expression. So for instance, CD90, a more stem-like marker, is really lighting up this particular cluster here, cluster 1, and you can see C14 and more spread expression across these different clusters. So that's nice. We can map, protein data onto cells, but where this gets really useful is we can say, well, what's the genotype of those cells, right? So our triple mutant here, our DNMT3, NPM1, FLT3, they were enriched in that stem-like community and the progenitor-like, whereas the NPM1 FLT3 that lacked the DNMT3 allele, they were spread out more differentiated. And that matches quite closely to the observation we had in mice, where having the early DNMT3 allele led to greater expansion of the stem cell compartment, and the NPM1 FLT3 alleles had more of a differentiated component. So we're quite excited to be able to use this platform to understand, if the observations we saw in mice were informing, or were being, re-identified in the human biology as well. So this is how we've used our models to understand clonal evolution in leukemia. We have, of course, these models in hand. We've looked at patient samples pre and post therapy, and I'll kind of give you a little snippet of how we've done that and how we are going to continue to do that to understand what's happening in patients, how we can model it back in mice. And I think these are kind of cool examples of this. On the left, we have a patient sample where the patient had a myeloproliferative neoplasia, MPN. And in the red bars here on the left, this is their MPN sample. And you can see the dominant clones are this CALR ASXL1 set of clones. This patient unfortunately transformed to AML. And when they transformed the AML, when we ran the Mission Bio assay, we found there was really one very large clone that was CALR ASXL1 mutant, and now it picked up an IDH1 mutation, right? So this is evidence of a largely monoclonal evolutionary event, right? Where when you're transformed from MPN to AML, it seems to be driven by a single monoclone. The patient on the right is an AML patient who had a FLT3 mutant leukemia. They were treated with a FLT3 inhibitor, right? So in red on the bars on the left, you can see that they have a large number of FLT3 mutations. When they were treated with a FLT3 inhibitor, we have our B sample in blue here. They did not recur with one single recurrent clone. They recurred with 4 or 5 different KRAS and RAS mutations, really having polyclonal resistance to this FLT3 inhibitor. And this gives you an idea of how different disease patterns or different patterns of clonal evolution can have monoclonal versus polyclonal patterns. We see this in the patient population, and we're using our tools now to try and see if we can model that in the mouse models in a more deterministic manner. So, to give you an idea of where we're going from here, there's been some really great work done in both the mouse modeling space, really revolutionizing how we model leukemia and how we model solid cancers, integrating CRISPR-Cas9. And that's something we're doing in the lab now, where we can start to have CRISPR laying down different clonal hematopoiesis events, and we can do multiplex genome editing to understand where those events are occurring, and then use single-cell DNA sequencing with Tapestry to go find which cells have those mutations and how they combine with our engineered mutations. We're able to take human samples, like the ones I've shown you, look to see which cells are actually mutated in those human samples, and make hypotheses about potential cells of origin in mice. And we can kind of do this loop where we set up models in mice to learn about the human system, characterize the human clinical specimen, and use that to drive hypotheses in mice to ask questions that are important for disease biology. I think the future of these assays really is in multi-omic assays. We need more material and more information about what these cells are. The DNA plus protein has been very powerful, and I'm quite excited about some work on Tapestry that's been done for DNA methylation with a CTM-seq. And we're certainly going to be looking at that with the engineered DNMT3 and IDH2 mutations that we've caused in these AMLs. And then really kind of bridging our, essentially what are structural variants with the with the CRE-DRE flip models onto the Tapestry platform, so we can have allele-specific PCRs that tell us whether we've, treated with flip, CRE, or DRE, and then combine that with your more traditional single nucleotide variation calls that occur on the Tapestry platform. And lastly, I'll end with a little bit of a plug for an R package that we've written for analyzing this data. We have had quite a lot of experience analyzing this in the human setting, and are growing our experience in using this in the preclinical modeling setting. I think this partners quite well with the Mission Bio Tapestry pipeline. You can take files from that pipeline, load them into this R package, and get to visualizations similar to the ones I've already shown you today. And we'll be maintaining this and working on this package the way forward. So we'd be happy to collaborate and answer any questions you have. So with that, I'd like to thank everyone who contributed to this work. Most of this done during my postdoc at Memorial Sloan Kettering with Ross Levine and my kind of scientific partner, Lindy Miles. And just to highlight a couple key collaborators here with Jen Trowbridge, now at University of Pennsylvania, has been a long-term collaborator, Martin Carroll. And then here's our lab at University of Pennsylvania. And, happy to tell you guys about our work. So thank you so much.
Thank you, Dr. Bowman, for that wonderful presentation and sharing your perspective on how cancer models can provide insights on leukemic transformation. So moving on to the next slide here, the first slide, I'd like to highlight how researchers from top academic and pharma institutions are leveraging single-cell DNA multiomics to validate disease models today, to understand how mutations in genes contribute to disease pathogenesis, how the preferential co-occurrence of these mutations can contribute to tumor fitness, spread, and treatment resistance, and identifying therapeutic vulnerabilities of these tumor populations that can be taken advantage of. Some other applications here include drug screening to monitor efficacy of novel therapeutics and augment existing therapies, preclinical studies to optimize treatment protocols, and finally, Tapestry can help shift the paradigm of healthcare by providing a comprehensive picture of the clonal landscape of individual patients in order to design a patient-tailored treatment approach. So moving on to the next slide. So why is it important to understand cancer at the single-cell level? Cancer is a very heterogeneous disease with multiple clones harboring multiple mutations. There is a complex pattern of cooperativity amongst these different mutations and a constant evolution of these clones as the disease progresses and upon treatment, as you can see from the time-lapsed fish plots on the right there. So it becomes imperative to understand these mutational patterns in clones. And as Dr. Bowman highlighted in his talks, the sequence of acquisition of mutations in tumors makes a difference too. So can we see these more deadly clones earlier and identify therapeutic vulnerabilities that we can target? These are all the questions that need to be answered to reach that goal of eradicating cancer, and Tapestry can help with that. So moving on to the next slide. So meet the Tapestry here. This is a novel multi-parametric platform that enables you to analyze SNVs, indels, CNVs, and surface protein within a single workflow to confidently validate disease models with single-cell resolution. You now have the option of multiplexing samples to conserve samples and improve throughput and reduce the cost per sample. And couple this with an easy-to-use software solution for data analysis. The platform provides a robust solution to validate cancer models and bridge the gap between discovery, preclinical, and clinical studies to accelerate drug development. So moving on to the next slide. How does our technology work? Based on a novel two-step microfluidic workflow, individual cells or nuclei undergo encapsulation to form isolated droplets that contain protease enzymes to allow access to genomic DNA. The lysate then undergoes a second encapsulation step where the contents are merged with barcoding beads, PCR primers, and reagents to target specific regions of the genome. Now, sequencing libraries are then prepared and sequenced on any Illumina platform. And for surface protein detection, antibodies targeting the surface protein are conjugated to oligos. There is a brief antibody staining protocol upfront of the cell encapsulation step. The antibodies are subsequently digested during the first encapsulation protocol, leaving behind the oligo barcode within the droplet. The oligo is then captured and integrated into the genotyping library and reported out as corresponding NGS reads after sequencing. Now, in addition to the easy-to-use instrumentation, we provide a suite of software support upfront. The custom panel design, where you can design your own panels, as well as a tapestry pipeline for multi-omic data analysis. So on the next slide, by linking genotype and immunophenotype information, we can holistically understand single-cell biology that bulk technologies cannot provide. In terms of genomic output, we are able to report out single-cell SNVs, CNVs, indels, loss of heterozygosity, and structural variants. And for the proteomic readout, we're able to report out single-cell surface expression for lineage marker identification and potential therapeutic antigens expressed in malignant cells, which has generated tremendous interest amongst the immunotherapy development community. Moving on to the next slide here. So just to highlight certain real-world cases, you're looking at publications highlighting the use of Tapestry for validating disease models, both in cellular and mouse models. You see here a recent publication from Dr. Eliza Ten Hacken, where they developed a model to identify loss-of-function mutations responsible for CLL transformation to Richter syndrome. And the other example on the right I'm showing here is a paper from Dr. Ilaria Jacopucci's lab, where they developed a preclinical model for identifying drivers of AEL. And these studies use multiplex CRISPR gene editing to generate mutations relevant to disease. Tapestry was able to analyze the efficiency of gene editing, both on and off edits, unravel the clonal diversity of disease drivers, and identify therapeutic vulnerabilities associated with the leukemia genotype. Now, on the next slide, we have publications where base editors were used to identify relevant disease drivers and variants responsible for therapeutic resistance, right? So on the left here, this wonderful paper from Dr. Benjamin Haley's lab at Genentech used a base editor to modify two genes, EGFR and BRAF, to identify single and compound variants that confer drug resistance in small cell lung cancer. Tapestry was able to verify the base edits by assessing amino acid changes, zygosity, and co-occurrence of alleles within the cell. The study is a great example of how Tapestry can enable the prediction of resistant mutations before drugs reach the clinical trial stage, so that the underlying evolutionary pathways can be halted. Moving on to the next slide. So this publication, it uses Tapestry for accelerating drug discovery. And on the left here, a wonderful publication from the lab of Dr. Andreeff, who used single-cell DNA sequencing to track mutational and immunophenotypic changes that could alter potency of a combinatorial treatment that they used in their study. A second example on the right is this outstanding publication from the Skorsky lab at Temple University that used Tapestry to design a patient-tailored approach based on the clonal landscape of individual patients to simultaneously target and eradicate all malignant AML clones in vivo PDX models. So I'd like to end with this slide, the very last one, on how you can gain access to Tapestry if you're looking to advance your cancer research and in drug development. We have customers who've adopted our platform in their labs and use our reagents and consumables upfront of their NGS system, and then use a Tapestry pipeline and insight software for data analysis and visualization. You can also take advantage of our pharma assay development services, where our in-house experts provide full end-to-end support on assay development and assay design, wet lab services, and bioinformatic analysis as well. And then we have our strategic alliances, where we collaborate with our partners to develop off-the-shelf solutions. So for more information on these offerings, please visit our website at missionbio.com or reach us at info@missionbio.com. Thank you very much.
Thank you, Dr. Robert Bowman, and thank you, Divya, for an outstanding presentation. We're going to open the floor up for some questions. So again, please submit your questions in the Q&A box, and we'll address them during this Q&A session. For those questions that we cannot get to today, I invite you to still email them to info@missionbio.com or into the chat box, and we'll address them offline after the webinar. So we have a few questions here. It looks like one for you, Divya. Can you talk about the value of single cell data on the tapestry versus methods, or traditional methods like Western blotting or bulk NGS, and kind of what are the benefits of single cell versus those orthogonal methods?
Thanks, Christy. But when you look at bulk methods like, bulk NGS, you're again looking at, an averaged out data, right? With single cell sequencing, it provides you that resolution and granularity where you can look at the co-occurrence of mutations, how do they come together to provide that added fitness and strength in order to, what is the clonal diversity of these mutations? It's questions like this that actually give you an idea of what is actually happening in your disease, and also being able to identify the order of acquisition of these mutations is what really gives you a deep understanding of how disease, the origin of disease and how it progresses. That really changes. Trying to understand that at that level of resolution can really help with changing treatment outcomes.
Thank you, Divya. And I think this kind of follows on to that question, but in a more preclinical setting, how do you think this data can help us better understand patient stratification methodologies?
Yeah, so I mentioned, one thing that you can do is trying to understand the actionable druggable targets, and how do they co-occur in these cells to bring about that added fitness. So what are all the druggable targets that you can identify, and in addition to mutations that might actually cause resistance to those treatments. And so by knowing that prospectively, you can really make the right choice of what treatment, how these patients are going to respond to those treatments. And I think that's going to really help with patient stratification.
Thank you, Divya. Okay, and I think this question is probably for Bobby. You discussed using single cell data to better understand the clonal evolution of mutations. I'm curious to understand if you've used or even your potential thoughts on using single cell data to assess different disease model types and their ability to accurately model human disease.
So how would we use the data to model different disease types, right? Like that's the kind of question. I think we're often interested in questions of how different mutations lead to different lineages of cells, right? So one thing we've seen in the patient data is able to use platforms like this to ask we can certainly deep sequence and find mutations. We can show the disease has perhaps a strong myeloid component, but understanding which mutations can kind of infiltrate different branches of differentiation is something you get from these assays. So that helps us distinguish things like clonal hematopoiesis that can broadly have alterations or broadly contribute somatic mutations to many different types of cells versus transforming mutations that may really restrict down to just the myeloid lineage. And so we can see that in the patient samples, and that's something that we try and model into the mice, and that getting the resolution on the different lineages is kind of where we're headed in that way.
Great, thank you. Can you talk about the applicability of single cell multiomics for the purpose of drug screening?
Yeah, that's interesting. So I like the Skorsky paper on this. They did a nice job on trying to understand. This is a study where they took different AML patient samples and treated them with drugs ex vivo and wanted to see how clones responded to different drug combinations. And this gives us a nice view into which mutations are most susceptible to different drugs in actual patient samples. I think that's hard to do with different methods where you're not sure which cells contain which mutation. So that's one nice way to do it with different manners of drug screening. In vivo, we can certainly look at single, not necessarily screening drugs, but screening how cells respond to a single drug, right? So you can set up a scenario where you have a multi-mutant leukemia, maybe experimentally, or in a PDX model where you have many different patient samples, and you can see how those samples respond to a given drug. But we've looked at that as well, and kind of other groups have, especially Koichi Takashi's group has looked at how patient-derived xenografts can respond to different therapies as well. So I think both those are quite nice ways to do this.
Sounds great. Thank you. And another question for Dr. Bowman. You mentioned a clonal expansion of many clones after FLT3 inhibitors. Did you see any of these clones before treatment? How deep did you sequence the pre-treatment sample, and what do you estimate your lower limit of detection is?
Yeah, so we've done this a couple different ways. I showed you an example today that had about 8,000 cells profiled, and we did not detect them prior to the inhibitor treatment in that particular sample. We've certainly tested other samples where we did detect the mutation. So I don't think de novo versus acquired resistance, that's not only one of those two things for RAS pathway mediated resistance to FLT3 inhibition. I think it's a really interesting question on what's the difference between patient outcome and patients going in that have either pre-drug treatment resistant clones versus only acquiring them after disease. So I think we see both on the Tapestry platform. Your resolution's how many cells you run. So it's between 10 to 20,000. It's 1 of 10 to 20,000. If you're a bit of a skeptic, you may say, I want 2 cells, right, or 3 cells to really believe it. And I think that's reasonable with any sort of error-corrected approach you use. So the lower limited detection is probably 1 in 10,000. And we've also done this with bulk error-corrected sequencing, and I think there you're reasonably looking at 1 in 10,000, maybe up to 1 in 20,000 again in the bulk then. And then other studies we've used ddPCR, where you can certainly get this down to 1 times 10 to the 5th or 1 times 10 to the -6. And we haven't done that for the FLT3 inhibitor treated samples yet, though.
Great. Thank you, Dr. Bowman. And I think this question is for Divya. Understanding next generation sequencing NGS for DNA mutation screening is straightforward, but could you please explain how it applies to proteomic analysis?
Next-gen sequencing for proteomic analysis using our platform?
Yes.
So with proteomic analysis, we currently are able to, look at surface antigens, right? And so as you can see from Dr. Bowman's presentation as well, it really helps you understand the cell, the lineages, and be able to track where these mutations, link genotype with phenotype, which is very important. And secondly, we also have surface antigens for various immunotherapies, to be able to identify, for example, in multiple myeloma, we have surface targets like BCMA and GPR5CD. And so what that will enable, drug trialists in immunotherapies is to be able to identify the expression of these markers beforehand, to see if their drug is going to be effective or not. So both looking at cell type lineages as well as identifying surface antigens for immunotherapies would be very useful in this perspective.
Perfect. Thank you, Divya. I think that was all the time we had for questions today. If we've missed anyone, we'll address your questions via email after the webinar. A very big thank you to Dr. Robert Bowman and Divya for a great presentation today. And we'll reach out with a link to the recording of a webinar within a week. So please stay tuned and have a great day. Thank you.
Thank you. Take care.